Okay, now we're going to conclude this section. So now you know why we study cancer, you know different definition of cancer depending on the perspective. You know that, for example, from a very general genetic perspective, chromosomal rearrangement is important. Also, those rearrangement and changes in the DNA cause a transformation in the cells, and we talk about the different stages of cancers. Yeah, that a primary tumor, a tumor that are in the circulatory system or circulatory tumors, and secondary tumors are different. They activate different genes, and the treatments can be also different. So now we're going to focus more on the genetic components, and we're going to talk about oncogenes. Yeah, what is an oncogene and why this is important, and then we will talk about tumor suppressors. So let's start. Okay, so now we're going to talk about oncogenes that produce unregulated cell division, and I want that you see these two graphs. Yeah, these two that they showed like uh, um, they try to make an analogy. Sorry, between a car and cell division and a proper cell division. So in a car you have a gas and brakes. So a gas is very important because you can go faster. Yeah, you need to move, and this is why cars are so important because it let us help us to move from one region to another one. But they also need a braking point, yeah, to stop in the part that you need to go. Yeah, in the same way, cell division require we or any tissue require that the cells divide, but they also require that you have breaks where you stop those cells, those tissues to from proliferating. Yeah, so there are two main components in this car: a gas and brakes. In a cell division, now seen from a perspective of cancer, you have two type of genes, and I will talk. There are four, but these one are two that are fundamental to understand cancer. One is proto-oncogenes that we just talked about them, and the other one is the tumor suppressors that are the brakes. These one are in charge of the proliferation, the proto-oncogene, and these one are in charge of inhibiting. If any of those genes is not working or both are not working, is when this uncontrollable cell division occur when we are not regulating properly the cell division. Okay, so here in the second schematic, you can see that the pro-oncogene turned into an oncogene, and now you have too much gas. In the case of cell division, you have too much proliferation, so you end up are going to break the car, okay, because you are not stopping at the right time. Okay, so although this is the brakes are there, you still have too much. You still can crash the car. And if both organ, you the tumor suppressor and the proto-oncogene or the gas, and there is no brake, things can get worse. And this is why there are different type of cancers, and this is why there are different speeds in which they can develop. So if you miss one, it can go at different speed, and if you miss the two of them, it can occur faster. Okay, so I'm going to summarize here. I have the summary. So remember, some proto-oncogenes work like an accelerator pedal in a car, pushing the cells to divide again and again. So we talk about that. And others work like a faulty brake in a car parked on a hill, also causing the cell to divide and check. Yeah, so again, two type of important genes that are regulating cell division, and both of them or both have a different function, but if both are unregulated, it will cause that the cell division is not regulated. Okay, now we're going to focus on the oncogenes. Okay, so uh, we're going to start talking about an oncogene. So what is an oncogene? And here I show you in the schematic. So there are two terms that you need to remember. One that is proto-oncogene and the other one that is an oncogene. And here you have an schematic. So the proto-oncogene is a gene where that promote proliferation in a regulated way. Yeah. However, when there is a mutation, this proto-oncogene, yeah, become into an oncogene. So now you have an unregulated mitosis. Yeah, and those changes are one of the early stages that lead to the appearance of cancer. Why? Because tumors start forming. Okay, so what is an oncogene? An oncogene is a mutated gene that has the potential to cause cancer. So before an oncogene becomes mutated, it is called a proto-oncogene, as I show you here, and it plays a role regulating normal cell division and cell death. And cancer can arise when a proto-oncogene is mutated, change it uh, from an oncogene, change it into an oncogene, sorry, and causing the cell to divide and multiply uncontrollable without any control. Okay, so easy. Yeah, so every time that one of the key steps of cancer is that the cell division is not controlled. Why? Because there is a mutation, as you can see here, and now something that it was pretty well regulated, like mitosis, now it can go out of control. Okay, now we're going to talk about the relationship between oncogene and viruses. And maybe some of you and some of you have already watched videos that I recommend you where they talk about oncogenes and viruses. Yeah, and so the first way that when we start finding that viruses can produce cancer, in some cases it was because they were attaching into regions, turning one proto-oncogene into an oncogene. Okay, so just from the beginning, the word oncogene suggests that they are causing cancer. And, and initially, oncogenes were identified in viruses that could cause cancer in animals. Later on, they found that those ones were mutated copies of normal cellular genes that so this is why they call it proto-oncogene, because they are before they're able to cause cancer. And as I said before, they just produce uncontrolled cell growth. Yeah, and they can escape from cell death, which might lead to the development of cancer. Okay, now let's talk about some examples of oncogene. There in the textbook you will find hundreds, and maybe there are more, and they depend on the context of the cellular environment. It's not as simple, but I'm going to, at least you need to remember these three. Yeah, so there are several hundreds of oncogenes that have been implicated in human cancer. And if you remember RAS, maybe you have read about RAS, you have watch some of the videos that I suggest. I'm going to show you a video in a second. And also some of the papers that you might be reading, they will talk about RAS. Okay, this is a very important gene involved in the production of cancer. So some people said that they are between, they are involved in 25 to 30% of the cancers, depending on the literature. So somebody that has cancer tumor, generally there was a mutation in RAS. Another one very important is MYC, also an oncogene, and this retinoblastoma that we will talk in the class about this, and all of them are an oncogene. Yeah, and so MYC is involved in, and we will talk about this, about all these different landmarks of cancer. Yeah, so one of them is that they are able to escape from cell death, they are produce genome instability, they produce induce angiogenesis, so new blood vessels to give food to the tumors. Yeah, yeah, they need what they need oxygen and nutrients, and this is brought to them through blood vessels, and there are a lot of things. So MEC is involved in many things, and retinoblastoma was found because it caused a disease in retina. Yeah, so people with mutations, they will have very particular abnormalities in the eyes. Okay, So we will talk more about that. Now I'm going to show you the video about RAS that I think is one of the most important that you need to know. Okay, so I'm going to show you a video of one of the most important oncogenes, and you will hear all the information about oncogene. I think I played that video in the previous, or not I play it. Is in the it's on Moodle in case that you wanted to watch it. But if there is something important of this class, is that you remember this gene. It is one of the most important genes involved in cancer. I think it's found in 30% of human cancer. So generally, it's one of the early stages. You say like, okay, one cancer appear. It's likely that one of the early stages is that there was a problem with this gene.
RAS is essentially the way that cells are constantly monitoring what's happening in the environment outside of them. And they're trying to make decisions as to whether they should divide, die, arrest, and not divide. The mutations that we see in cancer essentially leave the RAS protein switched on, but it can no longer be properly switched off. The easy way of thinking about it is that a cell is being told to grow, 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 grow all the time, even though there's nothing outside in the extracellular environment telling the cell to grow. If you take the most common of the RAS genes, which is KRAS, it's mutated in roughly a quarter of all cancers, for example, in 90% of pancreas cancer. It turns out to be a very difficult problem to make a drug to. I would venture to say that there will be a drug approved for a class of tumors with RAS gene mutations sometime in the next decade, maybe in 5 years.
Okay, so fascinating. Remember, you will see hundreds of papers that they will talk about this gene, maybe in the one of your projects, and they will be talking about RAS, or maybe in one of the projects or the paper that you already presented, cell biology or developmental biology, they were studying this gene because if it's a clear example of an oncogene in which if it's not working properly, you have a lot of cell division. So I remind you, is we start talking about cancer in terms of uncontrolled cell growth. Later, we start talking about cancer in terms of chromosomal rearrangement. We talk about viruses. We talk about transformation in when those viruses infect the cells. And now we are going into the molecular basis. Yeah, so where every time we zoom in and zoom in on each field, cell biology, cytology, now genetics and molecular biology are giving us a better understanding or helping us to zoom in what is happening. So we talk about proto-oncogenes, and in the next session we will talk about tumor suppressors. And there are four type of genes that it will be discussed. So remember, there are four ways in which we can regulate the cell cycle, that if we have mutation on those genes, it can lead to the formation of cancer. So one of them, the one that promotes tumor, is proto-oncogene. The one that inhibit tumors are the tumor suppressors. And we're going to talk about that in the following. And in the following section, later on, we will talk about genes regulating apoptosis. and other ones are the ones that are involved in DNA repair. Okay, so uh, now this in the following sections we will keep using this to talk about how can cancer appear. Okay, so we are done with this section. I hope that now you remember the four type of genes involved in cancer, and you already know about one that is very important, that is the oncogene. And the next section, we will talk about the tumor suppressors. Yeah, and I hope that you remember that RAS is very important, MYC and retinoblastoma are very important oncogenes, but there are many more. Okay, so uh, I will be talking about these guys in the next section, the tumor suppressor.